Because we'll follow what we do in the morning. We don't have questions, but if you have questions, please find Dr. Hody afterwards. So it's a pleasure to introduce my colleague, Dane Wittrup, as the next speaker. So Dane is the DOPS Professor of Chemical Engineering and Biological Engineering at MIT, and he's also the Associate Director of Koch Institute. Dane's research has been focused on developing protein engineering technology and apply it to the discovery of new biopharmaceuticals. In particular, the use of its surface display for directed evolution of protein stability, affinity, and specificity. So he will speak today on the synergism between antibody and T-cell-mediated cancer immunotherapy.
Thanks. So although our lab does a lot of protein engineering, as it turns out, the story I'm going to tell you today has very, very simplistic protein engineering, and the actual treatment itself is fairly simple. It's trying to figure out what it's doing that we've been spending time on. And the work that I'll tell you about bridges standard antibody therapy for cancer, just an antibody that binds to a tumor cell and drives suppression of its growth, with the predominantly T-cell-mediated therapeutic effects we've been hearing about mostly today. But first, I want to just have a couple of slides to say why I think this topic of this symposium, immunotherapy, is potentially the best hope for a durable and robust treatment for cancer. Given the genetic instabilities of cancer cells, there's pre-existing mutations within any given tumor that are very likely to drive resistance to the most cleverly sculpted tyrosine kinase inhibitors. And so, in fact, resistance to these so-called targeted therapeutics is essentially universal because, as in any classic Darwinian evolution case, the mutants that are resistant to it are already present in the tumor. So this high heterogeneity is a problem if you have one drug, if you have a single target that you're hitting. However, if you allow an adaptive immune response to adapt with the adaptation of the tumor, it can track that. And so the standard metric for most drugs has been median increase in survival, so looking at the midpoint. And of course, I'd say that any one of us or any of our loved ones, if they were being treated, we would certainly relish those small but significant, statistically significant improvements. But if you were to look in the not too much longer run, the endpoint is really still the same because of this emergence of resistance. And although things such as Provenge have been said to have marginal effects because of that small change at the midpoint, it's really this tail that's been referred to several times that is remarkable, and that's the first foot on the first rung of the ladder, understanding what causes that small percentage of patients to have durable responses and then increasing it. And even today, we're already seeing simply combining some of these therapeutics accomplishes that. So it should be well known to everybody that there's tremendous activity in antibody drug development. The antibodies we hear about today were antibodies that were going to bind to T cells and alter their behavior, but the great majority of antibodies that are in development bind to the tumor cells. And good paradigm examples of these would be Herceptin, Erbitux, Rituxan. These are antibodies that are sold at the billions of dollars per year level and have some therapeutic effect. The antibody engineers that work on these think of them as working in terms of 3 different potential mechanisms. Often, we antibody engineers start out trying to block some inappropriate growth signal. There's a lot of activity now in conjugating chemotherapeutics to antibodies to get more precise delivery of highly toxic payloads. But what the antibody engineers and clinicians eventually realized was that, in fact, antibodies were doing what antibodies do in the natural immune response, which was bridging an innate response from effector cells to the tumor cells, and that this was an important, significant, and perhaps required component of any effective antibody therapeutic. And so to understand how best to exploit this and to link it together with the types of T-cell-mediated therapies we're talking about today should allow the full power of the immune system to be exploited in these therapies. So the antibody engineer's worldview would be, I have an antibody and I'm going to bridge effector cells, predominantly natural killer cells are what are expected to be important, but perhaps also other Fc gamma receptor expressing cells such as neutrophils or macrophages to a tumor cell and drive antibody-directed cellular cytotoxicity, ADCC. Whereas the types of immunotherapy we've been hearing about mostly today take the point of view that it's really spurring the T-cell response to the tumor that could drive an adaptive response that could respond to changes in the tumor itself. And the ways to try and intervene in that, for example, would be addressing the T-cells with antibodies or cytokines, trying to feed vaccines to the antigen presentation system. But of course, there's extensive communication back and forth between these two, and the data I'm going to show you today is an example of that. Both from processing and presentation of antigen from cells killed by ADCC over towards T-cell mediated immunity, and also alteration of the tumor microenvironment and helping to draw in T-cells. But in addition, T-cells have a significant effect back in the other direction. Most antibodies and the antibodies that are now marketed and being used in the clinic were studied in nude mice that didn't have T cells. So this whole aspect of the efficacy of antibodies for the entire history of the field to date has not considered that communication. However, one question people had was maybe we can use IL-2 to boost the effectiveness of antibodies by stimulating natural killer cell mediated ADCC. And in cell culture, there's no doubt that that works, that adding IL-2 does give significantly greater cell killing by natural killer cells against opsonized tumor cells. And that was convincing enough to motivate about 15 or 20 phase 1 clinical trials combining IL-2 and antibodies, and several phase 2 clinical trials, all of which failed to show any efficacy. Combining Herceptin, Rituxan with IL-2, the standard Proleukin IL-2 that's been marketed since the late 80s, has no effect in the clinic. So we wanted to set up a mouse system to study this, and we had a choice of about one. In terms of a syngeneic mouse model and a fully wild-type mouse with a surface antigen and a paired antibody against that self-antigen, the B16 melanoma with the TA99 antibody is pretty much the whole show. You can construct models where you have exoantigens expressed and then transgenic mice that are tolerized against that antigen, and I'll show you data in one case for that as well. But so all of the data I'll be showing you today involves B16F10 as the model system. We inject about 1 million cells at time 0, wait about a week for the tumors to establish. Penetrance is about 100%. All the mice die. I'll show growth curves and Kaplan-Meier for these mice. If we treat just with mouse IL-2, and again, we're doing everything species-specific, so we use mouse IL-2, we use mouse isotypes. So IL-2 alone has no effect on the growth curve or the survival. The TA99 dosed once a week at 100 micrograms has no effect on survival. And combining the two really has no statistically significant effect either. So recapitulating the results seen in the clinic for combining IL-2 and an antibody, there was no effect in this model system. But we thought, IL-2 is cleared so quickly, and the effect of it is so brief over this several-week, month-long time period, surely having a more sustained signaling through IL-2 receptor would have a bigger effect. So what we did was we engineered a monovalent IL-2 Fc fusion where we introduced point mutations in the Fc of the mouse isotype so that it doesn't engage the Fc gamma receptors effectively. So this is just a pharmacokinetic lifetime extender. And I'll also show you data for an albumin fusion that does the same thing. Pegylating the IL-2, we expect, would do the exact same thing. So the idea is to allow us to dose IL-2 less frequently, but to get a sustained exposure to IL-2, might we see a different effect? And the answer is you see a qualitatively different effect. If you treat these melanomas with just the FcIL-2 alone, you get a little bit of a slowing of the growth curve and extension of the lifespan. Again, the antibody by itself isn't effective. But when you combine them, and this is a treatment that's just once a week of the antibody plus FcIL-2, for as long as we treated once a week, the tumors stopped growing. And that's been repeated now up to, I don't know, it might be about 100 mice that we've done this with. It's a robust effect. We stop the growth of these very aggressive melanomas simply by adding the long-lasting IL-2 to an antibody. And that's exciting for us because of the large number of anti-tumor antibodies that are on the market, and the fact that IL-2 itself is an approved drug, and pegylated IL-2 and human albumin fusions to IL-2 have been in clinical trials and shown to be safe and equally effective as Proleukin, even though they're not marketed. So the idea that combining these two simple treatments had that big an effect was interesting. So we wanted to delve into what might be going on in the tumor while we were treating it this way. First, just to generalize the result, here's the data showing that when we make a mouse albumin fusion to IL-2, the therapy is equally effective. So there's not some effect of a residual ADCC from the FC component of the fusion. Something worth thinking about given the recent realization that anti-CTLA-4 antibodies are actually Treg clearance antibodies. We then also used a different model system. These were MC38 mouse colon tumor cell lines that had been transfected to express human CEA and then put into mice that were transgenic for human CEA, so that was a self-antigen. And treating, again, these mice with that same treatment with an anti-CEA antibody, again, holds the line on the tumor growth. Once we stop after a month, and by the way, the mice don't lose weight, they're perfectly healthy as far as we can tell. Once we stop treatment, after another month, all the tumors grow out. So there are niches that repopulate the tumor. So this by itself is not a cure, it's just a very strong growth suppressor. And so to delve into what's going on inside these tumors as we're treating them, looking at the histochemistry, there's extensive infiltration of lymphocytes, and we've actually worked with Glenn Dranoff and his pathologist collaborator to look at a large number of slides to score them and try and understand what's going on from that data. But what we also did, because we used flow cytometry a good bit, was we disaggregated the tumors and used lineage-specific markers to see what types of cells were infiltrating these tumors under the combination therapy. And so looking at CD8 T cells, here the I is the FcIL-2 treatment, A is the antibody treatment, or the combo. And this is 3 days after the first treatment, so it'd be day 9, looking at the numbers of the different, and I'm going to show you several panels here. And these are the significant results, the statistically significant results. When treated with FcIL-2, the CD8 T cells do increase. The antibody alone does not increase the infiltrating CD8s. CD4 T cells are not really increased significantly, except a small effect from the antibody treatment. Tregs, again, no statistical significance. There's a trend. You can see that the 10 mice, the different symbols all trend upwards, but it's not a significant trend given the variability. Natural killer cell infiltration is increased by the FcIL-2 treatment, whether alone or with the antibodies, so it increased in both of those cases. Macrophages increased by the combination therapy, not significantly by the IL-2 alone. But alone amongst these different cell types we looked at, neutrophils were only increased by the combination therapy. So there was there was really no trend even, when treated with either the FcIL-2 or the antibody in neutrophil infiltrates, but combining the two gave an extensive infiltration of neutrophils. And this is something that we'll see, this is convergent with the importance of neutrophils in this therapeutic effect. Some more data I'll show. The FcIL-2 does activate the intratumoral T cells. So if we take those same T cells out of the tumors and stain for activation markers like CD25 or CD69, there's a significant increase in any treatment condition that uses the FcIL-2. So these extra T cells that are infiltrating the tumors are also activated. So then what we did was we took the tumors after treatment and disaggregated them and filtered and centrifuged out the cells and took the intratumoral fluid and subjected it to a multi-analyte ELISA and found that just about everything we measured went up under treatment. So the comparison case was this was 10 tumors treated by the combo therapy versus 10 tumors not treated. And what you find is that just about everything we assayed in this inflammatory panel here was up. And so this is a log scale, so this is tenfold here. And we'd heard a couple times today the expression cytokine release syndrome, or more colloquially or colorfully, a cytokine storm. And so those paradigm cytokines that people measure to look for a cytokine storm are by and large amongst this most enriched set of amplified cytokines in the combo-treated tumors. So in essence, we've got a localized cytokine storm that's been invoked by this treatment. And then this particular signal, which jumps out as about a 50-fold increase, is of a chemokine. It's the mouse homologue of CXCL2, and it's a powerful neutrophil attractant, which then, of course, dovetails with the observation that the combo therapy was the one of the three that actually did increase neutrophils significantly in the tumor. So then we went through a series of cell depletion experiments to see how different cell types were contributing to the therapeutic effect. And doing an antibody-mediated CD8 T cell depletion experiment, so here are the T cells and here's after we've depleted them, we found that in the absence of CD8 positive T cells, the therapy is completely inefficacious, just ablated entirely. So you absolutely have to have T cells, CD8 T cells present during the therapy to get an effect. Same thing with neutrophils. If you deplete neutrophils with an antibody depletion, you get no slowing down of the growth. In fact, you actually get a little acceleration of the growth of the tumor and no effect on survival. So the two cell types, as it turns out, that are absolutely required for this therapeutic effect are T cells and neutrophils, CD8 positive T cells and neutrophils. Looking at natural killer cells, they contribute, but not to the same extent as neutrophils. So here, depleting the natural killer cells, you can see that there is some holding of the line, but the survival is different, and it's an intermediate effect. The natural killer cells were contributing to the therapeutic effect, but were not required for the full effect. We did macrophage depletion using an anti-CSF1R antibody. We also did it with some data I'm not showing with anti-CSF1 antibody. And from those experiments, it would appear that macrophages are not contributing to any significant extent to the therapeutic effect. So of the possible Fc gamma receptor positive cells that could have been driving the antibody-mediated part of this therapy, neutrophils absolutely need them. Natural killer cells help. Macrophages don't need them. Depleting CD4 positive T cells, interestingly enough, actually improved the therapy. Of course, that would be depleting both helper T cells and Tregs, so it's not straightforward to interpret this. However, of that cohort, 3 of the 5 mice actually did have cures, which does suggest that immunosuppression in the tumor microenvironment by Tregs, when that was lifted, made the therapy all the more efficacious, which of course points us in directions for things to try. Trying to get at what the neutrophils might be contributing to the therapy, because neutrophils have many, many functionalities. A fairly straightforward one would be to say, are they using one of their main effector functions, which is generation of reactive oxygen species? And simply injecting luminol in a mouse, as it turns out, myeloperoxidase will generate a signal that you can read out on one of these instruments, these IVUS instruments. And so here's just with 1 mouse each. Here's an untreated and a treated mouse. And we're now repeating this with a larger cohort. The combo therapy with antibody plus FcIL-2 does drive the assayable presence of oxidative burst in the tumor. So as it turns out, I was then giving this an earlier version of this talk to the faculty lunch talk in the Koch, and Mike Yaffe, my colleague in the Koch, has been studying neutrophils and myeloperoxidase for years and had already upstairs in the mouse facility mice that had knockdowns of the myeloperoxidase. Perfect. So we took those mice, put the tumors into them, and looked to see what effect the combo therapy had, and it's intermediate. So this line here is the growth curve for the tumors in the myeloperoxidase knockout. And clearly, much like the natural killer cell result, they contribute. You need to have that myeloperoxidase function to get the full therapeutic effect. There's still some effect, statistically significant, different from no treatment, but a good fraction of the effect is actually coming from this oxidative burst. We saw interferon gamma was one of the many cytokines increased in the tumor upon this therapy, and so one of the reasons why B16F10 melanomas are not very immunogenic is they express very low levels of class 1 MHC on their surface. So this is immunohistochemistry staining for MHC. Under the combo therapy, those levels are increased. Under the combo therapy with interferon gamma depletion by adding an anti-interferon gamma antibody, that effect is diminished, in fact, below even the levels of MHC that were expressed in the untreated tumor. So this is one of the contributions that the interferon gamma is making the tumor cells more visible to a T cell response by expressing peptide MHC antigen on the surface. When we do that depletion with anti-interferon gamma antibody, again, we have an intermediate effect where there is some therapeutic slowing of the tumor growth curve, even in the presence of interferon gamma depletion, but it's clearly contributing. A bit surprisingly, and we've done this actually a couple times now, the TNF-alpha depletion antibody experiment showed that it was not contributing to the therapeutic effect, even though it is amplified, and a lot of cytokines are amplified in the tumors. The TNF-alpha blockage did not affect the therapeutic outcome there. So just as a cartoon to schematically represent the strongest results of the various different experiments there, the picture is that the dominant effect of the FcIL2 appears to be through its effect on the T cells, and I'll show you some data through the end of this talk confirming that, which then, by providing a higher interferon gamma in the tumor microenvironment, would have a pleiotropic effect. Lots of things will be happening. Tumor-resident T cells have been shown to make this chemokine, but it's possible also that the T cells are inducing other cell types to make that chemokine, such as mast cells or macrophages. The neutrophils then require, for their full effect, the ability to use that one part of their armamentarium, the myeloperoxidase. And then it's this collaboration between an innate response and the adaptive response that gives that strong month-long effect for as long, I say month-long because that's as long as we treated. We didn't extend it further. So let's look at T cells in a little more detail. What we did then was we did an adoptive cell therapy experiment where we took monoclonal T cells from a transgenic mouse that express a T cell receptor that recognizes a peptide antigen displayed on the surface of the melanoma. And so here the nomenclature is the same: antibody, FcIL2, FcIL2 plus antibody. And now the other thing about these mice that these T cells came from is that they express luciferase so that we could assay them by injecting the luciferin. And in the adoptive cell therapy treated mice that didn't receive either antibody or FcIL2, after a couple weeks, there's really not much in the way of T cells evident. Antibody treatment also didn't really have an effect. But the FcIL2 treatments, once a week, actually do extend the lifetime of these PMEL cells essentially indefinitely. So here's actually the quantification for 5 mice in each case. You can actually see the spike after an FcIL2 treatment. You see the T cells spike, and then treat again, you see them spike, spike. We've now stopped. This is in real time around here. It's now been, 10 days following that cessation of treatment, and they're still present. And so what are the therapeutic effects? And it turns out they're fairly strong. So just the FCIL2 treatment once a week allowed the P-mel cells to stop growth of the tumors. However, once we stopped treating with the FCIL2, every one of the tumors grew out. Just adding the antibody to the ACT, interestingly, had this little shoulder effect where there was an extension, a period of time where the tumors didn't grow, but eventually they all grew out as well. But it was the combo therapy plus the T cells that had a dramatic effect of all 5 of the mice were cured. When we then took 2 of those mice and re-challenged them with the B16F10, they rejected 100,000 cells injected into their other flank. And the vitiligo effect was mentioned. This is what these mice look like. They clearly have a strong immune response against the pigment-producing cells elsewhere in the body. So these are the mice that were cured by that combo therapy. And then this is just a comparison set. Age-matched because even the older black sex mice get a little bit grizzled. So just to see that you see a little bit of gray at the temples here, but this is a more dramatic effect. Okay, so to sum up what I told you about, taking a PK-extended form of IL-2 and combining it with an anti-tumor antibody gives a qualitatively bigger effect than either of the 2 agents alone. And short-lived IL-2, the naturally occurring form of IL-2, does not have this effect when combined with antibody, either in the clinic or in our mouse studies. And this simple injectable treatment once a week of PK-extended IL-2 plus an antibody was holding the line on the growth of this very aggressive tumor for a full month, allowing a window during which potentially other treatments could be inserted. And that's something that we're working on actively, is seeing if there are third agents that come together with this, like ACT, that would give a more durable cure. And in terms of the mechanism by which this therapy is working, it's the CD8 T cells and neutrophils that are absolutely required. NK cells contribute, and interferon gamma is important, and so is the myeloperoxidase expressed by the neutrophils. And then there's this combination with CD8 T cells that did lead to cures. And so to acknowledge the people who did the work, Annie Guy was a former grad student who first stumbled upon this observation of the combination effect. Eric Zhu has picked it up and pursued it with vigor and took most of the data that I showed you. Byron did the experiment with the MC38 CEA different tumor model. Carrie is a grad student in the lab working on T-cell therapies and adoptive T-cell therapies. We've, throughout the work, been having very useful and helpful discussions with Glenn Dranoff, and then also doing some of the immunohistochemistry. I've been working with him. Lou Weiner is using our therapy now on a HER2 transgenic mouse model with some early preliminary exciting confirming results. I showed you the collaborative effort with Mike Yaffe on the myeloperoxidase, and being next door to Daryl, I go in and bug him every time I walk by unless he closes his door, and his discussions have been really instrumental in thinking about this. And that's that.